right, welcome. Today we will be talking about energy, cellular energy. Okay, so today's study tip, flashcards should have a maximum of 15 words in order to be flashed. I would say even less if possible, right? The less, the better to promote memory. Study them in both ways. Don't just go through them constantly forwards, but make sure you go backwards as well. So you could give the term if you're reading the definition, or you could recall the definition if you're given the term, right? Go both ways. Also, there were studies that showed that the color yellow can promote memory. So maybe you'll want to get yellow flashcards or even yellow legal pads to make your notes on. Okay, cell respiration and metabolism, chapter 5. Now, I am making this as simplified as possible. You will need to memorize much more when you take upper level classes past this class, okay? But for this class, I thought about what do you absolutely need to know just for this class, and I boiled it down to that, okay? So, metabolism of nutrients. There are 3 major nutrients used by the body, which you already know because we talk about these all the time when you talk about nutrition, diet, etc. And so they are carbohydrates, lipids, and proteins. And a lot of people always talk about cutting carbs, but carbs or carbohydrates are always going to need to be the majority of your diet. But if you're taking in too many calories or you're not taking in enough protein, then you would need to reduce your carbs a little, right? But most people actually don't have a problem getting enough protein because plants, every plant that you eat is going to contain protein, of course some more than others, and then of course any dairy that you consume will also contain protein, in fact large amounts of protein, and so then if you eat meat or if you eat any type of meat substitute like soy products or pea protein or other types of products, that will give you even more protein, okay? So anyways, usually people get plenty of protein in their diet. They may or may not get enough iron or specific nutrients, but that's for another lecture. Okay, so metabolism is divided into 2 parts. Breaking down what you eat. So catabolism is the breakdown of nutrients to be able to produce ATP. And then of course you'll also use some of these, the molecules that you've broken down to be able to make new molecules. So we call those building blocks. And then anabolism is using energy to put things together. So using those building blocks and using that ATP to be able to build more muscle, for example. Carbohydrate catabolism. This is primarily talking about glucose, but we do have some other types of sugars, right? You saw that in the last lecture or two, and we will just focus on glucose. And the reason we're going to focus on glucose is because it is the primary set of pathways in the body. Most sugars, most anything you eat can be turned into glucose and then feed into the steps that we're going to look at. And even things like protein and fat, which we'll see at the end of this lecture, they even will feed into later parts of the pathway. So it really is the main pathway of energy production in the body. And so there's 3 major processes, and we call this aerobic respiration because it requires oxygen. And glycolysis is this first major set of steps. And in this part, you don't actually need the oxygen yet. So this is the common pathway that all organisms pretty much can perform glycolysis because it does not require oxygen, and it's fairly simple. Only a few enzymes required to do this. So this is pretty universal. And we'll see in a moment for examples of if you don't go through aerobic respiration, what does that look like? But let's first get the main pathway down. So after glycolysis, you'll go through another set of steps called the Krebs cycle, and I believe your book likes to call it the citric acid cycle. They're both correct and mean the same thing. Electron transport chain is the last set of steps, and that's where you get the majority of your ATP produced. And both the Krebs cycle and the electron transport chain require that you have oxygen. The oxygen is actually required for the electron transport chain itself, but if you don't have the oxygen for the chain, then the Krebs cycle begins to shut down as well. We'll look at that. And so for now, overview. So this molecule here should look familiar. This is your sugar, right? Your glucose, C6H12O6, glucose. Then you're going to have some oxygen from breathing, right? And then those will be transformed through a series of steps with lots of enzymes. And eventually, at the very end, after you go through all these processes, you'll end up with water, carbon dioxide, which you know you exhale as waste, and then you'll get your 36 ATP, and that's for every single molecule of glucose. And of course, also you know that as a byproduct of all these chemical reactions, you're going to release heat. Okay, so this is the overview of carbohydrate catabolism, the breakdown of glucose to give, the goal is to get all this ATP, okay? When you go through all the steps and you make this ATP, it will be called aerobic respiration. It will consist of glycolysis, Krebs cycle, and electron transport chain. So another way to look at this, another overview, is that you eat food, right? It goes from your stomach into your intestine, and from your intestine it gets absorbed, the glucose gets absorbed into your bloodstream. Okay, so the glucose from the digestive tract is absorbed into the bloodstream, and if you don't eat enough food, then your liver can also break down stored up. And look how this is called glycogen. Recall that glucose is stored in the form of glycogen. We learned that last time. And so the liver can break down its glycogen to release glucose into the blood as well. So whether it comes from the digestive tract or the liver, you have glucose in the blood that will be absorbed by all the different cells of your body, especially your skeletal muscles and your brain. And then once you absorb the glucose, it goes into the cell cytoplasm, right? The cytoplasm of the cell. And that's where the steps of glycolysis will take place. And the byproduct, or the product rather, is pyruvic acid. Pyruvic acid is slightly too acidic, it's slightly toxic to our cells. So you have to change pyruvic acid, and you do that either in a way that will go feed into the mitochondria and start the Krebs cycle and the electron transport chain. Or if you're working really hard and you don't have enough oxygen to go through the Krebs cycle and electron transport chain, then you can take that pyruvic acid and make it into lactic acid. And that's the burn you feel when you work out. You're familiar with lactic acid. So that's anaerobic metabolism because you didn't have the oxygen to go through the full set of steps. You took a shortcut to basically just to get rid of this toxic waste, pyruvic acid, and turn it into lactic acid. Now, later on, once you're not working out hard, but you're at rest, you have plenty of oxygen available, then your body will start to take that lactic acid and turn it actually back into pyruvic acid and send it through the full set of steps so you can get the rest of the energy, because you only get 2 ATP out of glycolysis, but glucose has the potential to give you 36, right? 36 ATP. And so, of course, we don't want to waste all of that ATP that we could have gotten. And so that's why we'll take the lactic acid back when we're able to go through the whole process. Okay, so this is a really nice overview of carbohydrate catabolism. Memorize this, be able to draw this, label all the parts, okay? You'll need to know some additional information on top of this, but this is a really great foundation to make sure you have at least have this memorized, and then you can start adding on more information to it, okay? So let's move on. So glycolysis, as I mentioned, it takes place inside of the cell, in the cytoplasm or cytosol, whichever you prefer to call it. Glucose, as you recall, has 6 carbons. It's that ring with 1 carbon hanging out above it. This is a little depiction of that. So the ring with 1 carbon above it. So 1, 2, 3, 4, 5, and 6 carbons. But it's going to be split in half to make 2 pyruvic acid molecules that are each 3 carbon long. And when you do that, you will also have this very special molecule called NAD plus come and take hydrogen, and this actually happens twice, so that's why it says 2 NAD plus. And so when it picks up hydrogen from these from glucose being split in half, then you form NADH, and then you also have an extra 2 protons. And this is we're going to see this again. This is going to play a major role in the electron transport chain. Okay, so pyruvic acid is produced and is then going to be processed and sent into the Krebs cycle. And as I mentioned, these NADH molecules and the protons, they will play a role in the electron transport chain. During this process, you produced 4 ATP, but in order to split this glucose in half, you had to invest 2. So you spent 2, so your net is 2. And there's images of this in your book if you want to see the details. The final product, what you need to memorize, the final product of glycolysis, 2 pyruvic acid molecules, 2 NADH, 2 protons, so this really should have a 2 in front of it like it does right here, and then 2 net ATP, okay? So there was no oxygen required for this glycolysis, right? This is what's actually happening when you have a glucose molecule and we invest in ATP to open up that ring. Then we invest another ATP to split it in half. And so now you have the same thing, 1/2 here, 1/2 here. And so each step happens twice after that, right? So that's why this funny diagram. And so then you end up with 2 pyruvic acid molecules. So something you should notice is that each of these steps of glycolysis, you have 1, 2, 3, 4, 5, 6, 7, 8, 9 steps of glycolysis, which each require a unique or different enzyme. So glycolysis, though we say it's simple, it's actually still quite complex, right? You have all these different products, like all these steps. And the whole reason to have little steps. To have this broken into all these little steps is to be able to control and safely release energy in a stepwise fashion so that the body can actually utilize this energy instead of having it all lost as like a big burst of heat. We do a bunch of little reactions that we can gain something from. And one of the primary things that we're taking as we split this are these hydrogen atoms. And that's going to provide us with a lot of energy, you'll see when we get to the electron transport chain. In addition, we also did get to make a couple of ATP for each half. But remember, we put in 2 ATP, so we only get out 2 net ATP. I am not asking you to memorize these names. I just want you to know what I wrote here in black, that there's 9 different enzymes, so that means there's 9 steps to glycolysis. It's the sequential breakdown of glucose into 2 molecules of pyruvic acid, and along the way you were able to make 2 NADH and 2 H+. Please make sure to put the 2 there. And then you also were able to gain 2 net ATP. Okay? So I know this looks really hard and overwhelming, but you're not going to worry about all that. Okay? All right, now this is one chemical reaction you actually do need to memorize, and you're going to see this in lab as well. So it's in your custom lab manual. And we call this the this enzyme here, LDH, that's responsible for taking pyruvic acid and making lactic acid, is called lactate dehydrogenase. We saw this name of this enzyme in the last lecture. So remember, when you're working really hard, you don't have enough oxygen to take pyruvic acid and go through the full set of steps to make all of your ATP. Instead, you just have to quickly convert pyruvic acid into lactic acid to keep your body safe, because pyruvic acid is toxic. Okay, so if no oxygen is available, pyruvic acid is made into lactic acid, and it does this by the enzyme LDH. Okay, this process is called anaerobic metabolism because you didn't have oxygen. You only get the 2 ATP that you made from glycolysis, so this is not preferred. This is not a good pathway. It exists only to protect yourself from pyruvic acid, and then you'll have to convert this back the other direction later on when you have enough oxygen, so that then you can get the rest of the energy out of this molecule. Okay, so again, LDH, lactic acid dehydrogenase. The other term for this is lactate dehydrogenase. For the purposes of our class, we'll treat lactate and lactic acid as the same thing. So make sure you've noticed that this is reversible. Once you are able to take pyruvic into the Krebs cycle, you will turn lactic acid back into pyruvic acid. Okay, so what is the Krebs cycle? It takes place in the matrix or very inside of the mitochondria, right? So the mitochondria has 2 membranes, this outer membrane and this inner membrane. And inside, the very, very inside, is called the matrix. Pyruvic acid will be turned into acetyl-CoA, and sometimes this is referred to as the intermediate step because it's an intermediate between glycolysis and the Krebs cycle. Once you make acetyl acid, acetyl-CoA, then it's able to go into the Krebs cycle or citric acid cycle. Okay, so every time you do this, you get 1 NADH. And remember, you split glucose in half, so you have 2 pyruvic acid molecules to turn into acetyl-CoA and then feed into this. So for each glucose, you're going to spin the wheel twice, right? The wheel being the Krebs cycle, okay? And there are 8 steps to the Krebs cycle. And again, we're not going to memorize these individual steps. You'll probably do that in another class later on. This process is still technically anaerobic. It does not require oxygen. However, it will stop or shut down if your electron transport chain shuts down. So the Krebs cycle products will give you the carbon dioxide that you know from exhaling. It'll give you 3 more of these electron carriers, NADH, super important. And now we get introduced to another electron carrier called FADH2. And he does the same thing as NADH. He carries these electrons to the electron transport chain. Sometimes these are referred to as coenzymes. I like to call them electron carriers because that's what they're doing. And it's a more helpful title to remember its function. Okay, so remember for each glucose molecule, you make 2 pyruvic acid. So this process is going to happen twice. So what's your actual product or outcome for each glucose molecule? 6 NADH and 2 FADH2s, right? So everything here gets doubled because you're going to go through it twice. So here's one cycle. So 1 pyruvic acid gets turned into acetyl-CoA. Then that acetyl-CoA goes into the citric acid cycle. And we call it the citric acid cycle because this first part of the wheel is a citric acid molecule. Notice that as you go through these steps, don't bother with these molecules, but as you go through it, you get your first electron carrier, right? NAD comes and takes 2 protons, 2 hydrogens rather, and you get the proton here, and then you took the electron, right? It's an electron carrier. So you're taking the electron from the hydrogen and you're leaving a proton. And we'll see those again, like I said, in the next step. But I want you to notice that as you go through all of these steps of the citric acid cycle, you make 1, 2, 3 NADH, and 1 FADH2. You also make an ATP right here. And you're going to do this twice because you have 2 molecules of pyruvic acid. Okay, I've been trying to be very repetitive because I know that this is a hard topic. So if you're not inside the matrix of the mitochondria, but rather if you're in these folds, then these folds are called crista. Crista is the individual, and if you are talking about all of plural, then multiple would be called cristae. Cristae is the plural form of crista. Okay. What else did I want to talk about here? Yeah, so notice you have your outer mitochondrial membrane. You have the inner mitochondrial membrane. You have the fold called the cristae or crista, right? And then the very inside called the matrix. And I'm pointing out all these parts because you will have to use that terminology with telling me how the electron transport system works. Okay, so for the electron transport chain, it is embedded in the inner mitochondrial membrane. And so the other way to say that it's found in the cristae of the mitochondria. But I like to say specifically the inner mitochondrial membrane. Oxygen's required for the electron transport chain. It's required because it's the final electron acceptor. So you can think of the electron transport chain, it's a transfer of electrons from one thing to another, just like hot potato. You keep passing the electron down until finally oxygen takes the electron and keeps it. Okay? So you do want to follow our protons. You want to follow the electrons as well. So hydrogen's been split. It's split into the proton and the electron. The electron gets carried down the chain until it gets dumped on oxygen. But then you also have these protons, and these protons get pumped from the matrix to the inner membrane space. And that's going to create this huge gradient where you have a lot of protons in the inner membrane space, and they want to diffuse back into the matrix, but the inner mitochondrial membrane stops them from diffusing. And so they're only allowed to diffuse back into the matrix when the ATP synthase enzyme opens up and allows it to come. And by opening up and allowing some of these protons to flow back into the matrix, this provides a strong burst of energy, strong enough to force a phosphate onto ADP, and that's how you make your ATP. So really, even though we talk about passing the electron and having a final electron acceptor, oxygen, the real power of the electron transport chain is coming from the proton gradient. Okay, so this is a really good video that goes through all of these steps. Make sure you watch this multiple times. Make sure you understand what we mean by the proton gradient, how is the gradient formed, and how is the proton gradient used to produce ATP. What do we mean by electron? What do we mean by final electron acceptor? So what is the electron transport chain, and how is that used to produce ATP? Okay, so make sure you watch these videos. Okay, so this electron carrier, NADH, comes to the first pump in the electron transport chain. It gives the hydrogen up, and that by doing that, you're going to give the electron to the pump. It's going to pass it down the chain, and the proton gets pumped all the way through into the intermembrane space. So you start creating this gradient, and you've passed the electron down the chain. The electron will get passed again to the second pump, and again to the third pump, until finally it is given to oxygen, and they'll form water. Okay. At each pump, you will also pump more protons across, creating more of a gradient of protons in the intermembrane space. When you get down to this huge protein here, this is the ATP synthase. You have built up a huge gradient of protons. It allows a couple to come back into the matrix. When it does that, you get a burst of energy, enough energy to slap on another phosphate group onto ADP, therefore making ATP. In this process, you also, remember, you let in a couple of protons. Those protons will be used. In fact, they will basically reunite the proton with the electron onto your oxygen, and that's how you make your water. So we temporarily separate the proton from the electron, and then we let them come back together. So that's it. That's the overview of the electron transport chain. By the time you get to this, you're bringing all those electrons from the Krebs cycle, from glycolysis, and you're bringing it to the electron transport chain. So you're going to do this over and over and over again until you make 32 to 34 ATP. Remember, you made some ATP 2 from glycolysis and another 2 from the Krebs cycle. And so then you'll make the rest here. So without the electron transport chain, you have very, very little ATP, right? And with it, you get a lot. And it's all due to this proton gradient, making a huge concentration of protons in the inner membrane space, so that when you allow a couple back in through ATP synthase, it's this big flow burst of energy. Okay, so in summary, you've passed the electron from protein to protein down the chain, and at the end it gets put with oxygen to make water. And glycolysis, Krebs cycle, and the electron transport chain all together give you a yield of 36 to 38 ATP. And this is the theoretical yield, but when you look at the actual system, in a living system, there's actually an energy cost. And so your actual yield is less. It's 30 to 32. Your book gives you a little explanation as to why that is. This is another one of the topics that you could talk about in your discussion to explain the difference between the theoretical and the actual ATP yield. It's one little paragraph in your book, and then you can practice explaining it in the discussion. Okay. And it's first come, first serve kind of thing, right? You cannot repeat topics that people have already posted. So everyone has to come up with something unique to talk about in their weekly discussions. Okay, so now we talked about how we break carbohydrates down to release energy. But what if you already have enough energy? What if you've eaten more calories than you are burning? Then you're going to build up glycogen, store up that extra glucose. And so carbohydrate anabolism is called, we can look at two forms. First, you have glycogenesis. And this is to make glycogen from excess glucose. And like I said, it's like if you eat a meal, but you're not actually using all of that energy at the moment, you'll store up glycogen. How do we do this? It's actually controlled by the hormone insulin. And glycogen is stored in the liver and the skeletal muscles. And this makes sense because your skeletal muscles are going to use the most glycogen. They need to have access to energy when you start exercising, or if you're running for your life or fighting for your life. You don't want your muscles to run out of energy, right? And then your liver is the only organ in your body that can release glucose into your bloodstream. So it needs to have glycogen in order to do that, right? So when glucose is all gone, you need a hormone called glucagon. Glucagon will stimulate glycogenolysis, which is the breakdown and release of glucose from glycogen. Okay, so to build it up, you need insulin, and to break it back down, you need glucagon. Okay, glycogenolysis, breakdown of glycogen. And as I mentioned, it's controlled by the hormone glucagon. Okay, so if your blood sugar gets really low, you've used up the glucose in your blood, then you're going to release glucagon, and that will signal the liver to break down glycogen and release glucose. So here, this image shows the relationship between building up glycogen and breaking it down. And so this here is a liver cell because only the liver can release glucose into the blood. Okay? So when you have extra glucose in the blood, you're going to have also insulin, and it's going to signal the production of glycogen. When your glucose levels drop, then glucagon is going to signal the breakdown of glycogen to make glucose and release it. Only the liver, okay? Only the liver can release glucose into the blood. If this was a skeletal muscle, you wouldn't have this part happening here, but you would be able to break down the glycogen and start glycolysis to supply energy to your muscles, right? So only the liver is able to take off this phosphate group and allow glucose to go out of the cell into the bloodstream. But if this was a muscle, then you would simply break down glycogen and feed it into glycolysis so that you can make ATP. Okay? So this is another one of those images that you need to make sure you can draw and explain. All right, lipid anabolism and lipogenesis. So to build up fats starting from sugar, you can take acetyl-CoA, which you remember is the molecule that feeds into the Krebs cycle. So after you've gone through glycolysis, you'll make acetyl-CoA, and that can be transformed into lipids. So you can make cholesterol, you can make a triglyceride, which is shown here, 1, 2, 3, triglyceride, and you can make free fatty acids as well. The term is lipogenesis, and your white adipose tissue is where the body will store most of your triglycerides, which is then used for making energy when you need it. Your brown adipose tissue, not really found in adults, mostly found in newborns, and it's used for generating heat. What else? That's it. Okay. So if you have excess fat and you're working out and you start breaking down fat for energy, this is called lipolysis, or the splitting of fat, right? And so then you're going to split it into its head, which is glycerol, and its free fatty acids. Glycerol can then be turned into pyruvic acid and then turned back into glucose so that you can use that glucose. You can use that glucose for energy, or you can use it as a building block, whatever the body needs. And the free fatty acids, same thing. You can turn fatty acids back into acetyl-CoA and back into glucose, and then go through the process, right, glycolysis, Krebs cycle, and etc. to make energy. Now, if you have a lot of this happening, so remember, ATP cannot be stored. It has to be used immediately by the body. And so if you already have enough energy, you have enough ATP, then you're not going to be able to feed all this glucose into glycolysis. And so what will happen is the excess lipolysis will result in what we call ketone bodies. And those ketone bodies, if they build up too high, can cause acidosis in the blood. And usually we only see that extreme amount, that extreme buildup of ketones only happens either if someone is starving, such as like the keto diet, or if you have diabetes type 1. Okay, so excess lipolysis results in increased fatty acids in the blood, and if the liver and skeletal muscles don't need to make more ATP, then those fatty acids will go through an alternative pathway which produces ketones, And if those ketones build up in the blood, it can make it acidic, and this can be very dangerous. Okay. Protein metabolism. Proteins are not used for energy. They are strictly used for their amino acids to make the proteins of the body that you need. Okay, so you always will use all of your sugars and then your fats, and the only time you start breaking down protein for energy is when you are starving, okay? Proteins are used to replace old amino acids, worn out proteins, and then any extra protein that you've consumed will be released in the urine as urea. So when you guys eat those protein bars and drink those protein shakes and you get these massive amounts of protein into your system, you're mostly just urinating that back out, okay? Amino acids can be turned into pyruvic acid and then go into lots of different pathways to make energy, so we'll see that. Okay, so this is probably the hardest diagram in your book, and it's also really important. So we're going to take a little bit of time. Sorry, when I pause, it's because I'm taking a drink. We're going to take some time looking at this image. So, as I mentioned, the liver can break down glycogen and release glucose into the blood. That glucose can then be used in glycolysis, and one of the intermediates could be taken to make glycerol, and that glycerol can be used to make fat, or it can continue and be made into pyruvic acid. If you don't have oxygen available, then it will be made into lactic acid. If you do have oxygen, then it can continue to go into the citric acid cycle, and then of course that can go into the electron transport chain, which is not shown in this image. But if you needed to make protein, you could take the pyruvic acid, you could take you could take pyruvic acid and make amino acids, or once you've gotten into the Krebs cycle, you could take one of those intermediates and also use that to make amino acids. If you do too much of that, you have extra amino acids, you will get rid of those as urea, right? You'll urinate that. Likewise, if you need to break protein down because you don't have enough energy to survive, then you can turn the amino acids into acetyl-CoA and feed that into the citric acid cycle and then electron transport chain. So yeah, take some time, follow the arrows, think about what it means, what's happening, where in the body is this happening. Okay, so only the liver can release glucose. These molecules can be turned into so many different things, right? Glucose can be turned into fat. It can be, essentially, it can be turned into protein, or it can be utilized for energy, right? After the Krebs cycle goes into the electron transport chain and produce ATP. Okay. Once pyruvic acid is turned into acetyl-CoA, and the reason that this is yellow, there's no going backwards. You cannot go from acetyl-CoA back to pyruvic acid. This is a permanent step, and that's because you removed the CO2 and you exhale the CO2. But acetyl-CoA can be used to make fatty acids or broken down to form ketones, right? Okay. Read the figure description in your book. Make sure you understand the parts of this figure. Okay, so the relative importance of the different molecules we've been talking about today between glucose, fatty acid, ketones, and lactic acid. Lactic acid, a little bit of it can be used by the liver and the heart for energy, but mostly it's not used for energy. It has to be turned back into pyruvic acid and go through the rest of metabolism to get energy from it. So it's mostly a loss. Ketone bodies, on the other hand, our brain, our muscles, a lot by our liver and a little by our heart. Everything can use ketone bodies. to get some energy. Fatty acids are full of lots and lots of energy. Our muscles love it, our liver loves it, our heart likes it a bit. Our brain does not like fatty acids. It will not use them. In fact, what our brain prefers is glucose. So another motivation not to go on the keto diet is that you need glucose in order for your brain to function, okay? Yes, some of the ketones will be used, but your brain prefers glucose, okay? This does not mean I am not telling you to eat sugar, okay? You should be eating whole grains, and you should be eating fruits and vegetables. Those are all going to be sources of glucose. Don't drink soda, don't drink other drinks, even juice. You want the fiber in the fruit for it to be healthy, okay? So watch your sugar intake. Don't just eat a lot of sugar, but get your good whole grains and fruits and vegetables. Okay, we're almost done. That is it. We are done. Yay, good job. All right, go eat.